Tom, I have a general question about the spectral reflect time. Symmetry allows to have not only vibrational state, which you detected, but also rotation. I didn't see any trace because in common molecule you have a vibrational level, rotational, and I don't see that rotational level should be out.
So the rotational spacing for a 1 micron or separation or 100 is kilometers. The B constant is just so small that the atoms don't live long enough to make a rotation. Molecules don't live long enough, and so we don't resolve it.
You don't have enough time to resolve it.
Yeah. That's
This not.
You said, I think, that you didn't have a microscopic model for the dephasing in the last part of the talk, but is it consistent with what you might estimate just from these molecules that you've studied, where a ground state atom comes through and just hits it?
I don't think so. We're making very, very few of these molecules, and so those molecule excitations, you're going to make. We're exciting for 1 microsecond, 2 microseconds or so. That is how long I did the molecule experiments, and I made like 1 or 2 molecules in my whole sample for the molecule experiment. Whereas in these Rydberg dressing experiments, EIT, I've got now my whole cloud. So if I had just 1 molecule in there, it's not moving very fast. So I don't think it would have much time to really come and have an effect that would be significant perturbation on the spectrum.
Well, but it's not the absolute number of molecules, but the number relative to the number of Rydberg atoms that you have. Right?
We have a lot of Rydberg atoms in there. Yeah. We can have excitations of 5–10% of the atoms are up in the Rydberg states. So these excitation fractions are getting up pretty.
So the dephasing rate you see is larger than what you would estimate.
Yeah, I think there are a couple things that we can think about. One could be is that there's no real decohering process at all. It's just that not only do I have an average density that's varying, but I have a stochastic variation in the separation, the atom-atom separations. So if I add that stochastic variation, I get atoms close together, further apart, and then if I add that kind of stochastic averaging as well as local density approximation, if you go through it all, that's another source of broadening on my apparent spectrum. So it could just be stochastic. It could be that essentially, this now gets a little off shakier ground, but you've just got a many-body system, and you're exciting up to this system where now your excited state is the excitation can kind of hop around to other atoms, and that just leads to decoherence of the spectrum. So you don't have to add it in the Hamiltonian, but in a many-body setting, you're going to get these decoherence processes. That seems to be Thomas Pohl's argues more for that as a source. He's working on a little better theory that goes beyond a single atom picture.
Questions?
So in the first part of your talk, where you had these measurements on the lifetime of Rydberg molecules, it seems that the decay rate scales linearly with the density.
Yep.
So is this a two-body collision of a non-Rydberg?
Those are good questions. Part of the simplest picture, the model I was put forward for the Stuttgart group, is that you have this molecule and another atom kind of comes and bumps into it. So there's kind of a geometric cross-section kind of thing. So a two-body collisional type thing. That can't quite be right because that would give the same loss rate for strontium as for rubidium. So that doesn't quite work. So I think there's still, so I think that the density dependence here is still somewhat of an open question. Somehow the molecules are more fragile. But you also, and another subtlety, so basically I'm just saying a big fancy way of hiding it. I think there are real subtleties on here. The way we detect these molecules, it's not just that the molecule has to be destroyed, because if I lose my molecular coherence and it goes to a different state, I'll still have the Rydberg atom there. And when I field ionize it, I'll still see the Rydberg atom. So really I have to have some process that de-excites the electron. And that's actually kind of hard to think about from on this time scale, or this energy scale for all the nearby electrons. But I think it's slightly more likely that these potentials are somehow pulling neutral atoms into the core, and that's somehow causing some loss of the Rydberg atom, destroying the Rydberg atom. That's what has to happen. But I don't have a great picture for it.
So you showed this data from the field ionization versus the voltage, basically. Can you use this as a pump and probe measurement of the spectrum of your Rydberg atoms? Like, let's say you excite first with a photon laser and then you do a probe. Did you test the time delay, let's say between field ionization and excitation of the Rydberg atoms.
Yeah, I mean, the lifetimes are all, and that is varying, where did it go? When we measure a lifetime, that's what we do. This is delaying between excitation and the field ionization. Okay? So yes, it is a pump, we wait, and then we probe. So it's lots of interesting things. Not overall, the signal goes down, so we're losing
Because when
atoms.
you show with that you have mixtures of higher electronic states.
That's what it suggests to me.
And that means that in typical calculation one should add the higher electronic states into the A functions. That we cannot assume that you just go to one potential basically, which is produce one mean field electronic potential. But
Yeah, I mean, you kind of expect this. When you put that pseudo potential of the ground state atom in the Rydberg function, you usually use the simplest approximation, you assume the Rydberg wave function is unchanged by the ground state atoms. But really, there's a little back action. So if you want to do it exactly, you'd say, well, having that atom there is actually going to change the Rydberg wave function a little bit. But traditionally, it's been neglected. But we're just so sensitive here. Well, I mean, the effect is big in the Strontium. So this is not a small effect, but it's 10% or so of the signal. So there's something going on. If it's not perturbation of the Rydberg wave function, there's something else interesting that we need to figure out.
We do Rydberg spectroscopy on molecules using chirp pulse microwaves. And there are a lot of advantages. One is we don't need to use ionization as a detection. We see free induction decay. And the experiment is also multiplexed. So if you chirp through many transitions, you see them all. And they're detected as free induction decay. And the atom would also do that. You could also go to higher angular momentum and make the lifetime longer. And so it's a completely new way of interrogating these systems. And it works extremely well. It's about 1 million times better for our experiment than using ionization detection. I really mean 1 million.
Yeah, no, I've seen some of those results for the millimeter wave spectroscopy of radicals. It's very impressive. We have to be able to put that apparatus on our ultra-cold apparatus.
But we also do this in a buffer gas cooled ablation cell. So we're talking about we're using Patterson and Doyle's idea, and so it's already cryogenic and it's also going to be very small.
Yeah,
And give you a completely different way of looking much more completely and accurately. We have 50 kilohertz resolution.
There is a problem though, because so far the density of these long-range Rydberg states has never been very high,
That's true.
which would make it very hard to see the FID signal.
But if you're looking at, I agree completely, but if you're looking at the loss of the atoms, you can, there's a lot of atoms there. And so if you can transfer these Rydberg molecules to higher L states, they will live much longer. And that is sort of extreme, too.
Yeah, that could be interesting to think about.
Now I have a completely separate question. Getting back to the molecule lifetimes. I know at least at one time the FAU group thought that the relatively accessible ion pair configuration in rubidium might play an important role in the decay dynamics. That would be very different in strontium. Is it possible that explains any of the differences you see?
Could be. I haven't looked at where the.
It's near 5x plus 8p in the rubidium.
To be honest, I haven't looked close. I haven't seen. Perhaps that is giving this extra. You're saying that could be giving this extra loss and dependence.
It could be a decay channel potentially.
Yeah, I don't know. That's an interesting possibility. And I don't know where or if there is a the strontium plus strontium minus curve.
I don't know where it is in strontium, except I'm sure it's different.
Yeah. That's something I should look into. Yeah, thank you.
You were saying that the lifetime of the triple D1 state strontium is relatively long, and that's convenient. It'll be longer in calcium. Why don't you do calcium?
Because I have an eye on quantum regenerative gas things, and it gets for other experiments, the calcium is much harder to get the quantum generous. So that's the main reason.
With that, join me in thanking Tom.